Hello, I'm Luca Comai, and we're talking about probability and how it applies to genetic analysis. Probability is the likelihood of getting what you want out of a number of possible events. And it can be exemplified by the toss of a dice. Say I want a 1. Oh, I got a 1. So what was the probability that I would get a 1? We can express it as a fraction. There are 6 possible outcomes. The one I wanted is 1 of the 6, so the fraction is 1 out of 6. We can also express it as a number, and in this case it would have been a number, it would have been 0.16. This is a number that will always be between 1 if you have absolute certainty and 0 if the probability is actually that you never get what you want. Last, we can express it as odds, and in this case the odds were 1 to 5. I'll tell you today about 3 methods. One is the multiplication method or rule, the other one is the addition rule, and the last one is the binomial expansion, and we'll see how these apply to genetics and what they mean. The multiplication rule. You multiply the probability of independent events. Consider, for example, a case where we have an heterozygote. It is a big A, little a. It has 2 alleles. When it makes the gametes, it can produce only 2 types of gametes, one that has the big A, the other one that has the little a. And so we know that each one of these gametes has a probability of 1 out of 2, or 0.5. If we consider the probability of getting an individual that has the little a genotype out of a selfing experiment, so we're fertilizing the flower of this individual with its own pollen. Alternatively, you can think of a cross where we have 2 individuals that have the same genotype. What is the probability of getting a little a individual? This is the classical place where we apply the multiplication rule. The 2 gametes are independent of each other, so we multiply the probability, and it is 1/2 times 1/2 equal 1/4. And in fact, you know that one way of getting this is to use a Punnett square. So what is a Punnett square? We draw a square, and we put all the possible contributions by the parents. So on the sperm side, we put it up here. We could have a big A and a little a. On the egg side, shown here, we could also have a big A and a little a. And you can see that the little a is 1 square out of 4, and that's where 1/4 is coming from as well. So a Punnett square is nothing but the representation of the probability field. The 4 square are all the possible events. The event we want is 1 out of 4. What I'm going to tell you is that the Punnett square is not going to be practical in many cases. And the case that I'm going to show you next would be a good example. Let's consider a case where we're looking at multiple genes, such as, for example, big A, big B, little b, big C, little c, big D, little d, and big E, little e. And now we are selfing this individual, and we're asking the question, what is the probability of getting an individual which is big A, little b, big C, little c, big D, and little e, little e? This is something which you could calculate with a Punnett square. It's going to be a pretty big Punnett square. There's actually a much easier way of doing it. That's the multiplication rule. So the way it works is that we calculate the probability of each gene, and then we multiply. These are all independent events, so the multiplication rule is perfectly suited for this calculation. So what is the probability of getting a big A, little a? We know that that is 1/4. Big B, a little b out of an heterozygous is also 1/4. The heterozygous output is 2/4. Big D is 1/4. And little e is 1/4. So we multiply all this, and we have 2 over 4 to the 5th power. This is so much simpler than running a Punnett square. We use the addition rule to calculate the probability of outcomes that can be produced by alternative events. And a good example is when we want a 1 and a 6 out of 2 tosses of a dice. Now I'm going to toss these dice twice. I could have 2 dice and toss them, it would be the same thing. So here I toss it once, I got a 3, so I'm already out of commission. But you realize that when I do this, there are 2 possible ways to get a 1 and a 6. I can get a 1 in the first toss and then a 6 in the next toss. Or alternatively, I can get a 6 in the first toss and a 1 in the next one. These are 2 different events which are alternative. Either one or the other happens, but both give me the same outcome, a 1 and a 6. So I can calculate the probability of this. This one is 1 out of 6 times 1 out of 6. This is for this particular event. And for this one, I get exactly the same probabilities. 1, 6 times 1, 6. This is 1 out of 36. This is also 1 out of 36. I have to add them because both of them will give me the result I want, and that addition will give me 2 out of 36, which I know is also equal to 1 out of 18. Where do we use the addition rule in genetics? The best example is the case where 2 heterozygous are mated, such as big A mated times big A little a. So these are 2 heterozygous individuals. Let's assume that this is the gene for albinism. It's a recessive condition that results when the recessive allele is homozygous. But what we want to determine is, out of this mating, what is the probability that we're going to get an individual which is big A, little a? Of course, you already know the answer, but let's consider how we got to that answer. We'll draw a Punnett square, and we populate it with the gametes. There are 2 types of sperm, 2 types of eggs. And let's consider how we got the heterozygous. There are exactly 2 ways, and there are alternative ways. First, we can get a little a egg fertilized by a big A sperm, or alternatively, we can get a big A egg fertilized by a little a sperm. These 2 events are alternative. They each have the probability of 1 out of 4. We add these 2 to give us the final probability, which is 1 out of half.